Hello, good morning all. Today is my first class, right? First discussion here, and today we will discuss about larval forms of crustaceans, right? So, crustacean is one of the important class of phylum Arthropoda, and phylum Arthropoda is the largest phylum among the animal kingdom, having the largest number of animals, that means insects, right? So let's start. Arthropoda is the special phylum which uh, represents frequent representation of development, two types of development, direct or epimorphic, or indirect or development by metamorphosis. This is very peculiar and this is very frequent type of development of phylum Arthropoda. And today we will discuss the larval forms of crustaceans, right? So see, these are some of the arthropodans. Examples like arachnida. These are crustaceans, right? These are insects. These are diplopoda and these are chilopoda. These are the classes of phylum Arthropoda. Now see this structure. This is shrimp, this is crab, lobster, barnacle, right? These all represent the crustaceans, right? See their structure. They show very peculiar type of structure, like the outer calcareous scales, then their antenna poda, right? So we will discuss one by one. See now the different larval forms of crustaceans. So what are those structures of larval forms? We all hear the terms like nauplius larva, metanauplius larva, Chiri's larva, etc. Right? So nauplius larva included order Decapoda. Metanauplius larva included order Nostostraca. Chiri's larva included Rhizocephala. Protozoa larva included Euphausiaceae. Joey larva included Decapoda. Elaima larva included Stomatopoda, and Megalopal larva included Decapoda. Mysis larva included Decapoda. Phylosoma larva included Decapoda order. And what are the significance of larval forms of crustaceans? Now see, larval forms represents a very different type of structure than the adult form. So it represents a very different peculiar type of structure other than the adult form. The larval form, passing through stages by stages, they grow to the adult form at the final stage. And the significance of larval forms, like they represent some phylogenetic significance of the line in their time of evolution, or many of the larval forms help in food gathering in their survival process, right? So these are some of the significance of the larval forms of crustaceans. Mainly, they represent the phylogenetic significance throughout the evolution. The larval form represents the phylogenetic similarities throughout the evolution to the ancestral form, right? So see the structure. This left one is nauplius larva and the right one represents the metanauplius larva. See here. And the basic important point, they use the median eye, which is the typical most important character of nauplius and metanauplius larva. Now these nauplius larva in many of the insects, converted by development to metanoplius larva. But in some insects, only one larval form still is there, that is nauplius larva, and in some insects, the larval form direct grow and develop to metanoplius larva, right? So the outer skeleton is called cephalothorax. They bears antenna, mandibles, rudimentary abdomen, maxillary bones, formal sense organ, median eye is very important, and the caudal fork is peculiar characteristics of metanoplius larva. Now, nauplius larva is found in cyclops and metanoplius larva is found in apus, right? See the next diagram. This is the protozoa larva of Euphausia and this is the zoe larva of crab. Now remember the term zoe larva. These represent the larval development of crab and this is one of the very important larval stage, right? Sorry. Dipios heart, carapace, antennulis, antenna mandibles and caudal forca. In case of crab, that means in case of zoe larva. But in case of protozoa larva of Euphausia, the last stages of the means this caudal stages of the larva is defined by telson. See the structure of telson, and one important structure of protozoa larva is uropod. Uropod and telson is the characteristics point of protozoa larva, whereas caudal forca represents the zoe larva of crab, right? See next, this is the mysis larva of Peneus and the right one is megalopa larva of crab. So we found till here that crab possesses two types of larva. One is zoe larva and next one is megalopa larva. See here, this is the zoe larva and this is the megalopa larva, right? And the peculiar structures in mysis larva of Peneus is uropod, we already learned it, and the megalopa larva possesses telson. Now see next the phylosoma larva of Palinurus. See the beautiful diagram of phylosoma larva of Palinurus. They possesses abdomen, head, maxillary pads, compound eyes, antenna, abdomen and thorax, whereas the cypress larva also possesses by lepus. See the structure of lepus and see the structure of cypress larva together. See the cypress larva, it possesses abdomen, anus, thoracic limbs, mouth, antennules, compound eyes and gut, whereas the Elaima larva of Squilla, the last structure in this page, see the diagram Elaima larva of Squilla which represented by telson, abdomen, carapace, compound eye, one sharp structure in place of mouth which is called rostrum, right? This is used for capturing food or to stick with somewhere, right? See the structure. This is what nauplius larva, microscopic nauplius larva. This is nauplius larva. See the diagram. This is the nauplius larva. We already studied here. This is the nauplius larva of cyclops, right? Antennules, median eyes, antenna, cephalothorax, mandibles, rudimentary abdomen, right? So this is the nauplius larva which is discovered by Muller in 18th century. The nauplius larva is the first fundamental stage in all crustaceans. Body is oval in shape and unsegmented with a large cephalothorax and rudimentary abdomen. There are three pairs of abdomens, namely antennules, antenna and mandibles, right? And the last two pairs of pyramous appendages used for swimming. There is a single median eye It has a well-developed digestive system for feeding on planktons, posterior and caudal fork. The nauplius larvae grow and undergo several molts and finally develop into next larva called meta-nauplius larva. See the structure. This is the meta-nauplius larva, right? See, this is the structure of meta-nauplius larva of Apus. This is median eye, frontal sense organs, antennules, antenna, maxilla, mandibles, maxillipeds, abdomen, caudal fork, right? So the characteristics of meta-nauplius larva includes in some nostostraca such as Apus, the nauplius larva transforms into meta-nauplius, which is slightly larger than nauplius and has cephalothorax and abdomen and a caudal furca. It also has a single median eye, antennulae uniramous or sensory, but antenna is large, biramous and locomotory in function. Mandibles reduces in size and are used for chewing food, right? See, this is structure of cyprinus larva. This is the structure of cyprinus larva, right? This is a cyprinus larva, the beautiful diagram, right? A barnacle cyprinus larva. This follows the metanauplius. It lacks a functional gut. Here the functional gut is absent. Now characteristics of cypris larva. Surplus ostracoda and Seripede. The nauplius larva changes into cypris larva. Body of cypris is enclosed into a bivalve cell which is secured by adductor muscles. There is one pair of compound eye. The antennulae is large and specially modified for attachment to substratum with cement gland. The second antenna is absent. There are 6 pairs of biramous thoracic appendages for swimming. The tip of abdomen bears a caudal furca, right? See, this is the protozoa larva of Euphorsia. See the beautiful diagram. The characteristics of protozoa larva. Metanauplius is succeeded by protozoa larva stage with 7 pairs of appendages. The dorsal shield or carapace has become enlarged and covered the dorsal surface anteriorly. 7 pair of appendages present in Metanauplius become well developed and capable of movement. Protozoa swims by the means of their antennae. See, this is the beautiful diagram of zoe larva, right? The zoe larva is the characteristics of crabs. Like many crustaceans have a larval stage known as zoe. At certain times of the year, it can dominate the samples. See the beautiful diagram of zoe larva, right? The characteristics is common larva of decapods having 1 pair of compound eyes. First and second maxillipeds are large and biramous. See here, the first and second maxillipeds are large and biramous, used for swimming, and abdomen is 6 segmented without appendages and has a caudal furca on the tip along with the telson. 6 pair of appendages along with caudal furca and telson, right? See, this is the Aloima larva of Squilla. This beautiful diagram of Aloima larva of Squilla. The characteristics are which hatch out from the egg, which is of modified zoe, means after zoe larva, the insect or the Aloima Squilla develop to Aloima larva. It is a pelagic larva having glass-like transparency. See here the glass-like transparency of the Aloima larva. A slender body with short, broad carapace and elongated form. The abdomen is segmented and has 4 pairs of appendages. See here, and end in telson, right? So this is the megalopa larva of crab. See the characteristics. In crab, zoe larva is transformed into megalopa larva which bears a large cephalothorax and small abdomen bearing small pleopods. Telson is very small. See here, the telson is very small. They have a pleopods, they have thoracic legs, cephalothorax, compound eyes, one pair of compound eyes, antenna, ciliate leg, antennulae and median spine. The cephalothorax is covered by brood carapace. See here, the outer skeleton is known as brood carapace. The first pair of thoracic leg is large. See here, the first pair of thoracic leg is very large and long and are used for crawling. See here, they are used for crawling. The first pair of thoracic legs, right? So this is the megalopa. This is the megalopa larva. Next we come to the mysis larva of Peneus. See the structure first, right? Then we go to the characteristics. Zoe larva develop to mysis larva. In Peneus, mysis larvae are elongated, laterally compressed and transparent. And anteriorly the carapace produced into spines and rostrum. See here the diagram. The 5 pair of cephalic appendages, 8 pair of thoracic appendages and 6 pair of abdominal appendages. See the diagram, right? And abdomen ends in telson, right? And uropod is also found here, right? And this is the phylosoma larva of Peleurus. See the structure first. It possesses antennulae, eyes, antenna, head, thorax, thoracic limb, abdomen, right? The peculiar structure, 3 pairs of thoracic appendages are very long. See here, the 3 pairs of thoracic appendages are very long and their tips are flattened oar-like for swimming near the surface of water. They are mainly used for swimming purpose. Now see the significance. They help in the dispersal of species, right? They help to study the different groups of crustacea. The larval stages are useful in finding the homologies and affinity of various groups. Means they are used in study of phylogenetic analysis, right? So today we can conclude our class like crustacea in their development undergo various larval stages with increasing complexity. As the Nauplius larva present in majority of crustacean life cycle, hence it is believed that all crustaceans might have evolved from the common ancestor which is resembled to Nauplius larva. The other larvae like Metanauplius, Mysis, Zoe, etc. show the stages of evolution of higher crustaceans from the Nauplius-like ancestor. See, these are the few diagrams of crustaceans, right? So, thank you till our next class. Bye.